Thank you very much, Lisa, and I'll add my kudos to your foundation for really doing a remarkable job of launching a foundation in such a short period of time, and I look forward to being associated with it and trying to help a little bit as we go along. I'll move very quickly through my talk, so hopefully we won't have a delay getting to mealtime. My mission today is to highlight for you some of the successes or some of the ongoing work that has shown the way, to some extent, with other ion channel disorders. CACNA1A is an ion channel. Hopefully you've received that message today many times. And disorders of ion channels, genetic disorders of ion channels, we collectively call channelopathies. So these are some disclosures. You'll hear me mention some of these companies. I'm not up here to advocate for any particular product. So what we know about channelopathies and their treatment can be broken down into really two broad categories. We've heard a lot about drugs. So pharmacological approaches have been the mainstay of treating these disorders. For rare disorders, though, we are always aspiring to find a drug that's already approved by the FDA that can be repurposed or prescribed for treating a rare disorder. But if there's not one, we need to develop a new drug. Emerging now, and a lot of excitement in academic labs, including my lab run by myself, a weird professor according to Dennis Lau, is excitement around new genetic tools and molecular tools that may enable a different classification of therapies. And there are many types, and I'll try to touch on some of these. Importantly, pharmacological approaches are the easiest ones. You take a pill. You can administer them orally. Genetic and molecular methods cannot be given by a pill, at least with current technologies. They have to be given other means, and for rare neurological disorders, that means direct injection into the nervous system, so non-trivial approaches, and that comes with other hazards. Another caveat here is pharmacological therapies we generally think of as symptomatic. They're treating the symptoms of the disease, seizures, migraine, hemiplegia, etc. The hope is that some of these emerging genetic and molecular therapeutic approaches will be disease-modifying. They'll have an opportunity to fix the disease for the long term. That's the goal, that's the dream. Now, we've heard a lot about gain and loss of function. I'm going to use that paradigm, oversimplification as it may be, around organizing how we present, or how I present, how you can use pharmacological or genetic techniques, tailored to what is the fundamental defect in the ion channel. So pharmacological approaches are pretty straightforward. If you have an overactive channel, you want to dial down its activity. We use a blocker to block its activity, or maybe an inhibitor. On the other hand, if it's a channel that has no activity, we want to revive it, we want to activate it. And in some cases, maybe that channel is too far gone. We can't revive it, but there's still a wild type copy of the channel that we can boost activity. So activators may be valuable. There are lots of caveats. Most importantly, what channel are you targeting, and can you be selective for that channel? CACNA1A is one of 10 different calcium channels in the human genome. Do we have a drug that can only target CACNA1A? Today, I don't believe so. Could we develop one? Sure, that's the goal. There are all kinds of other caveats too that make a drug worthwhile. Toxicity being one, whether that toxicity is brought about by targeting the channel itself or some off-target effect that we don't anticipate. And then for neurological diseases, the drug has to get into the brain. And most drugs don't get into the brain. They have to be built a certain way to get into the brain. Genetic approaches, same paradigm. Gain of function, overactive channel, we want to turn it down somehow with a molecular approach. Or correct the mutation. Loss of function, we want to turn on the other channel, the normal copy of the channel, so that it can replace the missing channel function. Again, caveats exist, all of which are the same except the first one here. Channel specificity is usually a little easier to accomplish because you're targeting the genetic code, which is more unique. but cell type specificity. Where in the brain do we want that molecule to go? Does it go throughout the brain? Should it just hit Purkinje neurons in the cerebellum? These are questions that have to be addressed, and that's where the big challenge is. Where do you put that molecular therapy, that genetic therapy? So these are big challenges in the field. Now, there's been lots of successes in the pharmacological treatment of channelopathies over the years. I've been studying channelopathies since, never mind, I won't tell you when I started, but when they started, I started working on them. And there's a panoply of sodium channelopathies that has attracted our attention over the years. And there are pharmacological solutions here for many of those. Many of these channel diseases are associated with gain of function, and therefore we turn to the medicine cabinet, and there is a lot of FDA-approved sodium channel blocking drugs. Some of your children may have been treated with some of these drugs. There are antiarrhythmic drugs to control heart rhythm. These have been particularly effective for treating cardiac channelopathies. Anticonvulsants that also block sodium channels have also been very valuable for treating some of the brain sodium channelopathies. None of these drugs are perfect. Why? First of all, they're not selective. They will block all sodium channels. That brings up a lot of issues of toxicity, particularly off-target toxicity, and so therefore there's been an effort in the last few years to develop more surgical, not surgery, but more targeted or elegant ways of modulating or blocking sodium channels. So a particular approach has been to either target a particular molecular defect or functional defect in sodium channels. And the example I'll show you is the idea of blocking what's called persistent sodium current. This is just a small abnormality that's been observed in many channelopathies. And a couple of examples here, both of which I'll talk about. Alternatively, how about blockers that don't block all sodium channels, that are more selective, that only block one or two different types? And I'll show you an example of one of those. And that's been a direction in the field. I think our lab was first to show that that selective block of persistent sodium current had anticonvulsant properties, and we did that. I did that in collaboration with Jennifer Kearney in my department and members of my laboratory, and used this investigational compound that was originally developed for the heart by Gilead Sciences, but they found that it got into the brain too well for it to be a realistic heart drug. So they gave it to us to test. In epilepsy, that drug known as GS967 was profoundly effective, particularly at suppressing persistent sodium current, an abnormality observed in some genetic mutations of sodium channels, also was present in a engineered mouse model of a mutant sodium channel. And it was much more effective than a conventional agent known as phenytoin. This is sort of the activity that we found at the level of the brain, and importantly, this improved survival of the animals, and so it looked very promising. We checked another animal model caused by a different sodium channel mutation and found the same thing, that we could rescue this fundamental defect in a more targeted way and improve survival of the animals. And this observation was replicated by another laboratory that was interested in the same mutation. At that point, though, the name of the drug changed. GS967 was the original name. It was licensed by Praxis Precision Medicines, a new company, and they changed the name to Prax 330, and that's why Prax 330 is in the title. However, the story continues. GS967 is what we would call a probe drug. It's only there to test biological effects in animal models and other things. It didn't have properties of a good drug. Therefore, Praxis re-engineered and began to do chemistry to try to find a better version, and they have come up with a better version that's called Prax 562. None of these drugs have advanced far enough to have a fancy name or a marketable name. But importantly, this drug, which also shows selective inhibition of persistent sodium current and efficacy in mouse models, they've actually now gone to the FDA and asked for a particular designation of this drug as an orphan designation drug. And that's important for rare diseases. The FDA has a fast track for drug development if you're targeting a rare disease. They give a certain number of tax credits to the company pushing this and some other opportunities to move it fast through the approval process. They're not trying to short-circuit any safety measures. They're just trying to move it forward as fast as they can. Praxis has plans, and I think they're planning to enroll sometime this year or early next year, patients with gain-of-function SCN2A variants in a trial with this drug. Now, that's the drug class to selectively block a specific mechanism. There are also now emerging selective channel blockers. Here's an example of one that only inhibits the sodium channel encoded by the SCN8A gene, known as NaV1.6. This was developed originally by Xenon Pharmaceuticals in Canada. They now are partnered with Neurocrine to move this ahead toward the clinic. And this is a drug that has really high selectivity for one sodium channel. However, importantly, in the lower graph on the right side illustrates that they can show this drug blocks SCN8A with a number of human disease mutations, except one, the big purple bar in the middle. That's a mutation that just so happens to be present at the binding site of the drug in the channel, so that the drug can't bind. So this is just one of the caveats, that you can develop a highly selective agent, but there's going to be some mutations for which it doesn't work. And only by profiling those mutations in an experimental system will you find that out. I can skip this. Backing up, the importance of me showing you that is, again, this compound shows a lot of promise in animal models, and therefore Neurocrine has gone forward and established and are enrolling in a study to evaluate it in children with an SCN8A form of epilepsy. The lesson here is that basic science discovery and working with investigational compounds and model systems can lead to clinical trials with an industry partner. Now, those are blockers. What about activators? There are loss-of-function sodium channel mutations. SCN1A, the cause of Dravet syndrome, is a great example of that. Are there ways to activate the channel? And just to show a spider to freak out Henry Colcraft, there is one found in a spider that modulates SCN1A sodium channels, shown here as labeled here as NaV1.1. But it changes the activity dramatically. And most of us thought, well, that'll never go anywhere. That would cause other problems. But apparently it doesn't, because now Steve Petrow's lab has demonstrated that this spider toxin, when given to a mouse, can actually rescue the mouse from seizures and premature death. How you get a toxin like that into the brain and get the FDA approved, I don't know. That's not been done yet. Maybe there's a hope for that. I'm going to move to potassium channels to show you some things that we've learned here. Just a little background. There's a neuronal potassium channel, well, two potassium channels known as CACNQ2 and Q3. They work together to create an important potassium current in the brain that controls electrical activity. CACNQ2 is the most widely studied of those genetically, and there's a spectrum of disease associated with early onset epilepsy with severe outcomes and some self-limited forms of the disease. And it turns out that most of the mutations are loss-of-function mutations. Same with CACNQ3, there's a spectrum of disease, but many of the mutations associated with epilepsy and neurodevelopmental disease are loss of function. So loss of function, we need an activator. So it turns out there was an activator already on the market called ezogabine or retigabine. The name is different, it's the same drug. It was marketed differently under different names to avoid confusion. But this was a drug approved in 2011. It was not marketed for the genetic forms of this disease. It was used for adults with partial onset seizures. It was approved, it was marketed, got a little bit of uptake, but then at some point the drug was withdrawn from the market, and I'll tell you in a minute why. But there was some investigational use of this for children with CACNQ2 mutations, and it showed some efficacy. The reason this drug was withdrawn in part was nobody wanted to use it because of stories like this, where people were turning blue. And I don't mean blue as in sad, I mean they were blue. Their fingernails, their eyes, their mouth, and their lips. So I think this is a byproduct of an unanticipated chemical reaction the drug had when exposed to sunlight in tissues, and so that diminished people's interest in that. However, Xenon Pharmaceuticals, I mentioned before, has now reformulated this for treating children, which it was not approved for before, and they've initiated and have gotten fast-track orphan status for this drug to treat children with this rare form of epilepsy due to KCNQ2. And they say this new formulation may protect the drug from this oxidation that occurs in light, but we'll determine that. Independently, Xenon has come up with another molecule for trialing adults with epilepsy using the same mechanism. And in another company, Biohaven has got the same thing. So this is, again, a target that was identified through genetics, and now there's multiple pharmaceutical companies interested in developing drugs for this. Those are activators. There are also potassium channel blockers that have shown some progress. There's been a lot of interest in a rare genetic epilepsy caused by this potassium channel gene, CACNT1, and this causes a severe form of early onset seizures known as malignant partial epilepsy of infancy. And the mutations almost invariably are gain of function. They're hyperactive potassium channels. And so the idea is, can we block them? Well, there were no existing FDA-approved drugs, so a repurposing notion came about, and Ethan Goldberg and David Beardham at CHOP decided that they would try a drug that had shown in the laboratory to block these channels, and that drug was quinidine, an FDA-approved drug that's been around for decades for treating cardiac disease and heart rhythm disorders. And this is the figure from their paper. It's hard to read, but they had a child that was having up to 20 seizures a day. They gave the kid quinidine, and after about 50 days, the kid went seizure-free. Remarkable. Published this paper, and everybody who treated a case with KCNT1 tried quinidine. Well, it turns out it didn't work as well as they thought. Here's another case series that some kids responded, some didn't. So really, we needed to get away from this anecdotal, one-off observations and do some kind of clinical trial. And so this was the first attempt to do a randomized clinical trial of quinidine for this disease. It was randomized, used a crossover design, which means everyone got either the drug or placebo, and then they switched, and then they analyzed the data. It was only 6 patients, but it didn't really work. And so now everyone's saying that was a false flag. So this is another lesson, that repurposing drugs, it's a very attractive area, and I'm not trying to say that it shouldn't be tried, but it has to be done right. This idea of an anecdotal observation leading to more anecdotal observations is just not the way to do things. A randomized clinical trial in some way needs to be tried, although it's very challenging with a rare disease with few patients available. I will show you a solution to that when I get to another part of the talk. However, it did inspire pharmaceutical companies and some academic labs to say, well, let's find a better KCNT1 blocker. So a group at Vanderbilt, led by my friend Dave Weaver, found a potential compound, and Praxis, again, interested in these rare epilepsies, have also identified a favorable compound. And so that's moving forward in the preclinical development. Another potassium channel blocker that some of you may have heard about is 4-aminopyridine, also sold in some places as Ampyra. This was repurposed. This is a drug that's approved for treating multiple sclerosis, and it was repurposed in a trial led by Holger Lerche in Germany for children with another form, a different form of rare potassium channelopathy caused by the gene CACNA1A. This is, again, was a gain-of-function potassium channel mutation for which blockers seem to be indicated. It turned out in the laboratory 4-aminopyridine, or 4AP as we call it, was very effective at blocking mutant channels and restoring the activity toward normal. So they did a clinical trial, and they found really a surprising and very effective use of this drug in that case. So most of the enrolled patients improved, whether they were old or young. One got worse, but most of them got better. And so this is quite an accomplishment. I think this is still a relatively new observation. I don't know what the next step is, but presumably this could be a treatment. Now, as I was looking into this, I wanted to see, well, what about CACNA1A and 4-aminopyridine? And so it turned out I tracked down, this was the original study that trialed that compound in children that had episodic ataxia and CACNA1A mutations. And this I show because of the way they tested it. They used a strategy for their clinical trial called a randomized double-blind crossover trial. But this is, in essence, an N-of-1 study. Has anyone heard of N-of-1 studies? This is what's being put forward as ideal for rare diseases, where you don't have huge populations that you can divide into a test group and a placebo group. This takes advantage of the fact that you can randomize treatment of an individual between the drug and a placebo and going back and forth. This is so-called crossover design. And this was done very effectively here. They only had 10 subjects, 7 of whom had CACNA1A mutations, and they showed efficacy in a great way. There was another trial that was done. It doesn't look like it's ever been published in a peer-reviewed journal, but a larger trial was done and showed efficacy as well. So I'm bringing this up only to illustrate this idea of rare disease, a need for clever clinical trial designs, and I think this N-of-1 type of randomized crossover design is worth considering. Now, that's the pharmacological side. The genetic side, genetic therapies, is where everyone is very excited about. These have the potential for disease-modifying effects, which is what everyone wants. We probably aren't going to ever get rid of symptomatic pharmacological treatment, but everyone is looking forward to the day when, like in spinal muscular atrophy, we can give a drug as in the form of an antisense oligonucleotide or a viral-delivered form of the same to really cure. I almost hate to use the C word, but to virtually change the course of the disease in the life of the individual. There are a number of forms that genetic and molecular therapeutics take. Here's a summary of that. If it was a overactive gene, a gain of function, then one wants to take that copy of the gene and knock it down, reduce its activity at the molecular level. The tools that everyone is interested in are so-called antisense oligonucleotides. That name comes from the fact that they are short, synthetic pieces of DNA. That's where the oligonucleotide comes from. Antisense refers to the fact that the sequence of DNA is complementary to the mRNA that it's targeting, and it will bind and degrade. There's examples of that I'll show you. There is also now emerging strategies that use the CRISPR system to try to downregulate how the cell turns on a gene. I haven't been able to find examples that were ion channels in that, but that's another approach. For loss of function, there's a lot more theoretical approaches. The classic is just what we call gene therapy. Okay, someone has a bad copy of CACNA1A, let's just give them a good copy. We'll get it into the brain somehow, and that will restore the function. That's the concept. Easier said than done, especially for large genes like CACNA1A. It's just so big, it's hard to deliver. So gene delivery is ultimately the challenge. But there's now emerging a lot of other strategies that are simpler that may enhance translation of RNA into protein or turn on the generation of RNA from that gene. So enhancing translation can be done a number of ways. I'll show you some clever examples that might be worth considering in CACNA1A. In particular, an approach to suppress what are called poison exons, parts of the gene that when incorporated into the messenger RNA, kill it. And there's a strategy now for SCN1A that's in clinical trials to try to suppress that. On the transcriptional activation side, I'll show you an example of a transcriptional activator that's an engineered transcription factor that's also now entering a clinical trial phase. And ultimately, all the hype is about, well, let's just correct the mistake. Let's gene edit. Let's edit the mistake right out of the patient. And this is where the the technology around CRISPR gene editing has come in. CRISPR gene editing is moving into the clinic in some diseases, but not neurological diseases. And there are two other derivatives of that. One's called base editing and one called prime editing that I'll tell you about. Well, I know a lot of you have heard the term antisense oligonucleotide, or ASO therapy, and this is sort of the concept of that. So the first image that you see on this slide is the dogma of molecular biology. DNA encodes the instruction set. The cell makes a copy of that in a slightly different molecule, messenger RNA, that's exported out of the nucleus of the cell into the cytoplasm, where it's then converted to protein. Antisense oligonucleotides, or at least one embodiment of this technology, it targets that messenger RNA with a short span of DNA and makes, instead of a single strand, which is RNA, they make a double-stranded copy, and that gets degraded in the cell by an enzyme called RNase H. You degrade the messenger RNA, the protein never gets made. So this is a great way of kind of turning off a gene. The usual strategy is to do something, to make something called a gapmer. You may hear that term. That's a jargon for a design of an oligonucleotide in which you have a central area of DNA, single-stranded DNA, flanked by RNA, and this is just simply to enable the process. One big question comes up always, which is, do you make this allele specific? In other words, your child has a particular mutation in CACNA1A, and it's a gain of function. Can I just get rid of that by targeting that mutation? That turns out to be easier said than done. It's very hard to discriminate between the two copies of the gene based on a small change. So therefore, most of the successes have been with allele non-specific, where they just target the gene and not try to discriminate the mutant copy from the wild-type copy. And it has been successful. One area where that's been tried is CACNT1. I mentioned that a minute ago in the pharmacological strategy section. But this is the gene with the gain of function. Can we downregulate it at the messenger RNA level with an antisense oligonucleotide? This is some images from a preprint from Steve Petrow's lab, where they developed first a mouse with a gain of function mutation, which has poor survival and epilepsy. And then they developed an antisense oligonucleotide, which doesn't discriminate between the mutant and the wild-type allele. It just knocks the whole gene down, and they can actually show fewer seizures and improved survival. This is, again, this is a DNA drug that is injected into the spinal canal of these mice to try to get, or into the cerebral fluid in the brain to try to get it to slosh around in the brain to have its effect. And it seems very effective. Another place where this has worked is with SCN2A, a sodium channel in which there are mutations that are known to be gain of function. Again, work from Steve Petrow's lab showed that they could develop a mouse with a recurrent gain of function mutation. The mice have poor survival, terrible seizures, and again, an antisense oligonucleotide targeting the gene indiscriminately, regardless of which mutant or wild-type allele it's targeting, was very effective at reducing the overall expression of this sodium channel in the mouse, and had and improved the lifespan and reduced the seizure burden. Same approach was used by another study by Miriam Meisler, University of Michigan, where they targeted a different sodium channel, SCN8A. Same concept. This was a gain-of-function mutation. They had a mouse model. They targeted the gene, indiscriminate, not allele-specific, and they improved survival and reduced the seizure burden. And then they started thinking, you know, there's another way that this could help a totally different epilepsy, which was Dravet syndrome, related to loss-of-function mutations in SCN8 and SCN1A. And the reason for that was from prior studies showing that if you attenuate SCN8A activity, those animals got better. So they repurposed in their own laboratory the SCN8A antisense oligonucleotide and showed that it actually improved the survival of Dravet syndrome mice as well. So this is a very curious example of where you get some collateral benefit from developing a therapeutic targeting one gene. Now, I'm not going to show you a lot of examples of gene delivery because there aren't a lot in the channelopathies. But I did want to share with you some terms related to the viral vectors. So gene delivery in the current technology uses viruses to deliver a gene to a target organ. The most popular right now, the one that's moving fastest toward the clinic, is a virus called adeno-associated virus. This is a virus that's not pathogenic for humans, doesn't make us sick, as far as we know. It seems pretty safe. It also is great for long-term expression in different tissues. And so it's conceivably a way of doing a one and done. You give it once, and that's it. You don't need to give it again because it gets into the cell and it stays there. The downside is it's easy to fill it up with DNA. It doesn't carry a lot of DNA. If your gene of interest is bigger than 4,700 nucleotides long, it's too big. CACNA1A is 6,000 or 7,000. So not possible to deliver in the usual way. There are also people that may be already immune to it for some reason, and so there's some downsides. Another viral vector, which has been around for a very long time, has really had trouble over the years, adenovirus, has some advantages and disadvantages. Main advantage is it can hold a bigger piece of DNA. It might hold CACNA1A, I'm not sure. But it doesn't last forever. It's very short-lived, and if you try to give it again, the first dose has basically immunized the person, and therefore they will have antibodies against it. So it's not great. There are also some safety concerns based on earlier work with it, and so probably not ideal. People have been lately talking about canine adenovirus. I must admit, I don't know much about it, but it can actually hold a large piece of DNA. It seems to incite less inflammation, and therefore might be able to be safer and be given more than once. I think it's unclear yet, and so those are the main ones that people are using. Lentivirus is being used for ex vivo gene therapy, where they take out cells, infect them, and put them back. This is probably not something for the brain. There have been two examples of using adenoviruses for delivering SCN1A, which is a very large gene, effectively into the brain of mouse models. One of those papers was published last year. The other one is, I think, just as a preprint that just came out last month. Another approach, other than gene delivery, also uses antisense oligonucleotides to boost expression. And this is one that I think has gotten a lot of attention, and this is this idea of poison exons. So poison exons are basically small pieces of DNA code that, if incorporated into messenger RNA, they would kill the messenger RNA, and you would end up not having a functional protein at the end. So one idea would be, can we target those? Can we use an antisense oligonucleotide to prevent them from being incorporated? And that will improve the efficiency of making the protein. Well, that's a strategy that seems to work. Lori Isom and her team, in collaboration with Stoke Therapeutics, tested this in a mouse model of Dravet syndrome and found that they could actually improve protein levels of the protein in the brain, improve survival of the mice, and reduce their seizure burden. And based largely on that preclinical work plus other unpublished data, Stoke Therapeutics, which touts this technology of poison exon targeting, they are now enrolling in a clinical study, and they've actually reported intermittently the interim results last year, a success in getting this new DNA drug into patients and showing some efficacy. So it's still early, but I think they'll probably pivot to a late-stage clinical trial in the near future. So there's hope for this technology. Then lastly, there are some more exotic approaches. This is one in particular in which they're targeting the same gene, SCN1A, with the goal of boosting expression of the gene to restore or to compensate for the loss of function mutations. This is a little more exotic, a little more complicated. Here they're engineering what's called a transcription factor. This is a protein that will bind to the gene in the nucleus of the cell and turn it on and generate lots of messenger RNA, which will turn into more protein and correct the defect. This is by a company on the West Coast called Encoded Therapeutics, and they did a collaboration with Jennifer Kearney in my department to demonstrate the efficacy of this engineered transcription factor to boost levels of SCN1A and reduce the seizure burden. And this, again, has moved forward to a clinical trial designated by the FDA as a rare orphan disease therapeutic, and they are now enrolling Dravet syndrome cases in this clinical trial. You hear a lot about CRISPR these days. There's one embodiment of the CRISPR technology that may be suited for loss of function mutations, and that is referred to as CRISPR-A or CRISPR activation. This is similar to what I just showed you, and it's a strategy to bring an artificial transcription factor to the gene of interest to boost its activity. This, again, has been tested by a group in Italy for SCN1A and Dravet syndrome, and have shown efficacy in boosting its expression both in neurons and in a mouse. And I think that I don't have the mouse data. Same technology has also been used for a different sodium channel, SCN2A, again published only as a preprint right now online, showing that it works as well in cells as well as in an animal model, and reduces seizure burden and I don't know if they only check seizure burden. So lastly, the hope ultimately is, can we just repair the mutation some way permanently and be done with it? That's the dream. I think that dream may come true. I don't know when. We're all hoping that something related to the CRISPR system is going to do that. But just so you have the big picture, CRISPR is this molecular trick that you can do in cells to modify their DNA. The goal, if you want to repair a mutation, is illustrated here, which is CRISPR itself, coupled with its enzyme Cas9, cuts DNA at the location near the mutation. Then you supply a donor DNA, which gets incorporated by cells' repair mechanisms, and you repair the defect. And that's the goal. And that can work. It has some liabilities, but that's ultimately the goal. One of the liabilities are on-target toxicities, which is if the cell doesn't repair that cut, you may end up with a screwed up gene, and you have damage, not repair. So that's the problem. So people are working hard to enhance the probability of it repairing, not damaging. But then you have a lot of concerns about off-target edits. What if you hit a different gene accidentally? So this is not a mature technology yet, but there's a lot of worldwide interest in improving this technology. Another idea, which was developed by David Liu at the Broad Institute, someone that Jen Pan knows, is base editing. Whereas CRISPR is trying to just cut and paste DNA into the genome, base editing is essentially correcting a typo. Okay, you've got a mutation, single letter is wrong. Can we just find an enzyme that converts one letter to another letter? In fact, those enzymes were known. David Liu's lab adapted CRISPR technology to target a spot in the genome, but drag along these enzymes that could then just literally correct the mistake. It doesn't cut the DNA on both strands. It has fewer liabilities that CRISPR has. There's still some concerns about it, but it seems like something with a lot of promise. And then the latest invention by David Liu's lab is called prime editing, and that's related. You use the CRISPR technology to target a spot in the DNA, but then you actually repair right on site. Instead of using an enzyme to convert one nucleotide to another enzymatically or chemically, here you just sort of rewrite that spot in the genome to correct it. And this shows some promise, and it's been shown to work in a mouse with progeria. So this is sort of the comparison, the pros and cons of these different approaches. I don't want to dwell on these, but the key liability of CRISPR technology is it has to cut DNA. And anytime you cut DNA, you risk having it misrepaired at the end, and that could cause more problems. Base editing sounds very promising, but not all mutations are amenable to that. Some conversions just don't work, and there's a possibility of having what's called a bystander mutation, where something is changed that you didn't intend to be changed. Prime editing, still pretty new. It's a very complex technique. I think it will be challenging to see how that gets delivered in an intact organism. So just some final thoughts, and I'm done. Clearly, CACNA1A as a channelopathy is amenable to a lot of different ways of treating it. Pharmacological primarily are going to be symptomatic, but the newer genetic and molecular approaches have hope of being disease-modifying. You got to have a model system, some kind of cellular or animal model system. And my last comment is just simply, these last comments are about watching out for uncontrolled drug repurposing studies. Anecdotal studies have not really shown very long-term durability. That kind of study needs to be done right in a controlled way. And then you've got to partner with the biopharmaceutical industry. These are things that, I'm sorry, my little lab at Northwestern can't do. A consortium of labs maybe would have a little bit more success de-risking, but the companies have to be involved. So anyway, thank you very much for your invitation, and I'm happy to ask questions.
All right, thank you, Dr. George. We just had one quick question regarding the use of 4-aminopyridine for epilepsy, because some parents were told or were thinking that that wasn't effective. It was like an earlier slide.
The slide I showed was using 4-aminopyridine for people with CACNA1A mutations, so a different disease.
Okay. That could have been the source of confusion. Okay. I'm going to start here.
So my daughter, who has CACNA1A and episodic ataxia type 2, was put on 4-AP about 10 years ago, and it was a complete game changer. She went from having 1 to 4 migraine/episodes a week throughout her childhood to completely getting rid of them. In those 10 years, she's probably had about 4 episodes with migraines, but they've been related to not taking her medication or heat and not taking her medication. So it's just a complete game changer for her. My question for you, though, is so 4-AP, you said, works for gain of function for CACNA2. Can I assume she's maybe gain of function then, based on that?
Yeah, so again, I wouldn't try to mix the two genes.
Okay.
So certainly CACNA2 is a completely different gene than CACNA1A. And there, the idea is that if it's a gain of function, 4-AP would suppress the overactive potassium channel. CACNA1A, actually I'm not sure what the mechanism is. I don't know that 4-AP directly targets the calcium channel encoded by CACNA1A. So I'm not sure.
Why it worked.
Yeah, maybe we don't care, but
Right.
yeah, but I'm sure there's a reason. It may be, again, it's just like any other drug. It may have benefits, not necessarily because it's targeting the over or underactive calcium channel.
Okay, and also she was misdiagnosed with epilepsy for 20 years, but it turns out she never did have it, and you cannot use 4-AP if you do have seizures. It does cause seizures. So at least that's my knowledge, or my
Yeah, it's remarkable that the drug is given to people knowing how it affects animals, because if you give enough in an animal, they'll have a seizure. So it has to be dosed right.
Does anyone else have questions for? Oh,
Right behind you.
Behind you.
Related to hers, and you just answered part of it. Yeah, my son actually was diagnosed with that CACNA2 potassium channel mutation, as well as he has the CACNA1A. And what I was told about the potassium channel diagnosis was, like 6 years later, it's by the way, millions of other people have this, it's benign, it's not really causing the seizures and the problems that your son is having. And then later he was diagnosed with the CACNA1A. And of course, we were told he was the first and only case ever. And so I'm realizing as I'm watching your presentations that he may have both.
Well,
And
yeah, I think what you said, I would need to know more, but I think if someone said that millions of people have this CACNA2, I'm filling in the blanks here, variant, that might have been a common variant that is probably not pathogenic. Just the way you said it makes me think that's probably the case, and so it was not thought to be the cause. Certainly something that has millions of people with it is likely not the cause, but I would have to know more about that.
Thank you, Al. That was a tour de force summarizing 30 years of ion channel drug discovery, therapeutics. Thank you very much for that. It was a really great overview. But of course, we always want more, right? I mean, so you've given us a fantastic overview, so now we're going to drill into the details, and vice versa. If you've given us a very specific thing, we would have wanted an overview. So drilling into the details, you were saying about CACNA1A, and we really have no good sort of molecules around for it, which is pretty much the case. Obviously, what Henry showed earlier was the agatoxin from the funnel spider. Do you think, putting aside the large molecule into the central nervous system delivery caveat, do you think that might be an option, at least for gain of function, potentially in these early days of, doing the experimentation towards whether these kinds of blockers might work, for instance, in an in vitro test system?
Yeah, no, it's a good question. So, I think a lot of people have been, over the years, very excited about channel selective toxins, neurotoxins, that maybe have therapeutic benefit. I don't know of an example where that's been translated effectively, but I do know people have thought a lot about it. And certainly, look, we do give toxins to people. There's this thing called Botox, which, it is a toxin. It's a little bit more, it's a little different, but I don't know of an example where one of these peptide toxins have been effectively given. I won't rule it out. I think there's still people actively trying to do that. On the other side, I think knowing how that toxin blocks might inspire designing a small molecule that could be a pill to do the same thing. So I think there's effort to do that.
Thanks, Al. Yeah, I second what Damien said. That was really beautiful and didactic, and I learned a lot from it. So one thing that I found surprising, I think we've been talking about both gain of function and loss of function, causing disease phenotypes. When you use an antisense RNA, DNA, that just gets rid of the protein. It's hard to imagine how that becomes therapeutic, because on the face of it, that should be like a loss of function.
Yeah, so I think you're right. If you're using an antisense oligonucleotide, using that first example where you're really just trying to down-regulate the amount of messenger RNA in a cell, yeah, it's very dose-dependent. If you give too much, you may convert them over to a loss of function situation, which may be bad. So these will be dosed in a way that will be very tricky, and so I think that's where the art of doing the clinical trial and the dose response has to be done. But I agree with you that you have to be extraordinarily careful. I think the same goes true for drugs, though. If you're trying to block the calcium channel, and you block it too much, you may have a different problem. So I think it all comes down to very, very careful dosing and to work that out in clinical trials.
All right, thank you. Does anybody else have any questions at the moment? If not, again, we encourage you to keep talking during lunch, and we will see you back at 2:30, I believe, correct? Okay, thank you again, Dr. George. Have a good break and lunch.